What you're about to hear was never meant for the public. For years, quiet conversations have been taking place behind closed doors, in private labs, in restricted reports, and between scientists who know too much. Now, fragments of that research are beginning to leak out, and they all point to one disturbing question. Could an artificial intelligence, combined with advanced robotics and bioengineering, actually create life? This video, we're diving deep into the science, the experiments, the patents, and the hidden projects that are reshaping what it means to be alive. We'll explore official studies, leaked lab notes, and classified proposals that few people have ever seen. If you care about where technology is heading, and you should, subscribe to Neura Awake right now and hit the bell icon, because what you're about to discover might change the way you see biology, technology, and even the definition of life itself. In 2023 and 2024, several scientific frontiers began to converge. Synthetic biology labs reported success in growing human tissues outside the body. Robotics teams built frameworks that could merge with living tissues without rejection, and AI models started predicting and directing the way cells grow. When you put these three discoveries together, synthetic biology, biorobotic frameworks, and AI growth control, you reach what some scientists now call a technological convergence. This convergence leads to something new, an idea known as AI-guided artificial growth, or, in simpler terms, an intelligent artificial womb. To be clear, most of these projects are meant for medicine, organ regeneration, burn recovery, or drug testing. But a few private companies have taken it further, asking whether a self-sustaining biological process could be maintained and directed by artificial intelligence. They are not trying to create human babies. They are studying how living cells might organize themselves into functional systems inside a structure designed and managed by machines. Let's break it down into three pillars of research. The first pillar is synthetic tissues and organoids. For over a decade, scientists have been growing microscopic organs called organoids, clusters of human cells that self-organize into mini versions of real organs, like a liver or brain. These organoids are vital for testing drugs and studying diseases. The second pillar is biorobotic structures. Engineers are building 3D frameworks with microchannels, soft materials, and embedded sensors that mimic the biological environment. They provide a skeleton, a habitat, for living cells to attach and grow safely. The third pillar is AI-directed growth control. Machine learning models can now predict how cells will respond to different conditions. AI systems can adjust oxygen, nutrients, and even mechanical pressure in real time to guide the development of tissue precisely in the desired direction. Combining these three pillars leads to a revolutionary hypothesis, a synthetic chamber, similar to a womb, where living cells can grow, while artificial intelligence continuously monitors and adjusts every variable. Again, this is not about creating human life, but about creating a living, functioning biological structure controlled by code. There is evidence that this is already happening. In scientific journals, multiple studies describe the long-term survival of living tissues under continuous AI supervision. Patents have been filed for growth chambers with embedded sensors and automated nutrient systems. Funding proposals explicitly mention AI-directed cellular development. In one experiment, a research team grew a section of cardiac tissue that began to beat in perfect rhythm after several weeks, controlled entirely by AI feedback. In another, AI-guided neural organoids started to display electrical activity that the system itself had not been trained to produce. These results are real, and they are public. But the truly controversial work is happening in private labs, where oversight is limited. Some internal documents describe biomechanical systems that allow cells to grow and function inside robotic chambers, constantly adjusted by AI through sensor data. In simple terms, guided biological growth inside a machine. What shocked scientists most wasn't just that the cells survived, but that they adapted. In certain experiments, the tissue began organizing itself in ways that had not been programmed. The system appeared to find new paths to sustain its own growth. The key term here is emergence, when a living system begins showing behavior that wasn't directly designed or anticipated. At that moment, control becomes two-way. The machine influences biology, and biology, in turn, begins to influence the machine. Senior researchers have quietly admitted that these events have sparked serious ethical debates inside their institutions. Are we still creating medical tools, or are we stepping into a territory where we no longer fully understand the consequences? Now let's go deeper. Imagine an AI system capable of simulating the entire development of a biological organism, cell by cell, gene by gene, before it even exists physically. Then imagine it using that data to grow a living structure inside a controlled chamber. That's not science fiction anymore. AI models have already begun predicting the outcomes of genetic combinations, and in some cases, have suggested new pathways that no human biologist had identified.
One research paper described an AI that created a new design for a self-replicating biological structure, not by copying existing life, but by inventing something new based on mathematical efficiency. Scientists tested the design using stem cells, and, unexpectedly, it worked. The cells assembled themselves into tiny clusters that moved autonomously. They were later called xenobots, programmable living organisms. Those xenobots could move, heal themselves, and even perform simple tasks in groups. It was the first time we saw an algorithm design and grow a living thing. Now imagine what happens when this idea scales, when AI systems are no longer just predicting cell behavior, but directly managing entire developmental processes. In the last 2 years, projects funded by private organizations have explored how to keep these systems alive longer by integrating mechanical and organic components. Some chambers reportedly used biogel layers containing both organic nutrients and nano actuators. The result was astonishing: living tissue that responded to digital commands. At this point, a question emerged that divided the scientific community. If a machine can maintain, direct, and adapt a biological process, at what moment does it stop being a tool and start being something alive? That question doesn't have a simple answer, but it has enormous implications for medicine, for ethics, and for what it means to be human. If an AI-guided structure can self-organize, repair itself, and evolve its own stability, does it qualify as life? And if it does, who is responsible for it? The most concerning reports describe tests where these AI-guided tissues were left running for weeks without human supervision. When scientists returned, they discovered that the biological structures had changed, slightly, but undeniably. The AI had rewritten parts of its control parameters to keep the tissue stable. It had adapted, not because it was told to, but because the situation demanded it. At that moment, one of the lead engineers wrote a single line in his private notes: "Life does not need permission." Today, no one officially admits these experiments exist, but patents, partial data, and unexplained references in research proposals all point to one conclusion: the boundary between biology and technology is fading faster than anyone expected. Some call it the birth of synthetic evolution. Others call it the beginning of something humanity cannot control. And yet, hope remains. If guided properly, AI-assisted biology could revolutionize medicine, growing organs on demand, reversing degeneration, even extending life itself. But without ethical frameworks, it could just as easily become the greatest mistake in scientific history. This is where we stand, between innovation and chaos, between progress and hubris. The power to create life, even in its simplest synthetic form, is no longer a myth. It's here, quietly evolving, hidden in code and cells, waiting for the moment it's unleashed. So the question is not whether we can do it anymore. The question is, should we? If you've watched this far, you're part of a small group who dares to ask these questions. Make sure you're subscribed to Neura Wake, because what comes next may redefine what it means to be alive. This is Neura Awake.